I'm working on.
Yes, that's fine. Just put it in presentation mode.
Okay, so thank you for the invitation.
Yeah.
I have
So
heard lots of excellent presentation, but my presentation will be a little bit different.
Sorry for the
We are
interruption.
aware of the complexity of living organisms. We know that.
Doctor, sorry for the interruption. Can you please put your presentation slides in a presentation mode?
Can you hear?
Slides are not in presentation mode.
It is in a presentation mode.
But for us, it is not. Fine then.
Cannot see.
Go ahead. Yes, go ahead, doctor.
Okay, so you can see. So we know that how complex a living organism, and one single cell, which size is about 40 micrometer, and there is 1.8 meters like DNA in a nucleus with 3.2 billion base pair. There are over 2,000 biochemical reactions in one single cell, and we know it's the DNA copy about 25,000 genes. But we also see that each biochemical reactions and the gene expression, gene expression is well organized, and they are harmonized to each other. We are talking about molecular biology, and we believe, and this is a target, that we believe that this system is organized on a molecular level. And I say that it's impossible that that complex system would be organized and harmonized with a huge molecules. We say that there is a submolecular regulatory system, and this is what I'm going to talking about. When we are drinking a glass of water, considering that there is a heavy hydrogen deuterium, we are drinking 3 different water molecules. H2,
Dr. Gábor,
yes?
Please, can you just put on your presentation? It's not moving.
I don't know why not. I share it.
Yeah, share and put it in presentation mode if you don't mind, please.
Is it not in a sharing mode?
Yes, now it's in a sharing mode. Yes, thank you.
Okay. Okay. So we are drinking when I'm drinking a glass of water, we are drinking 3 water molecules, H2O, H2O, or D2O. We know since the deuterium was discovered that due to the 100% mass difference, the heavy hydrogen behave differently in chemical reaction. So these H bond breaks much faster. And this is called isotopic effect, and this is well known for almost 100 years. And the heavy water was tested for 60 years, and it was clear when we increased the D2O concentration, for example, watering the plant, a plant grow watering with normal water. As the D2O concentration increased, the plants were very short and were not able to grow up. When we check the ratio in a normal surface water between hydrogen and deuterium, we can see it's 6,600 to 1, which means 150 ppm the D concentration. But in concentration in molarity, we can see it's about 16.8 millimole, the D concentration. It means that in a living organism or in a human body, the deuterium concentration is 30 millimole. And if you compare it to calcium, magnesium level, you can see it's pretty relevant. And in spite of the fact that the deuterium concentration is 6 times higher than calcium, nobody investigated the possible role of deuterium. So my study, when I started, the question was very simple: whether the life during the billions of years give any role of deuterium in the living organism. We can get the answer for this question if we reduce the deuterium and we see whether a deuterium depleted water causes any changes in the living organism. Our very first paper was published in 1993 in the Phelps Letters, and we said that the nature occurring deuterium is essential for the normal growth rate. What we found, we prepared media. This is 150 ppm normal D concentration, and we prepared media with 30 ppm. And we found that the cell growth was inhibited at 30 ppm, but the cell growth was stimulated over the natural concentration 300 and 600 ppm. So in that study, we also could prove that when a mice was transplanted with breast cancer, and the treated mice consuming deuterium-depleted water, at the end of the study, we had 60% of the animals, they were healthy, while almost all the mice had been died in a control group, which suggested that the cancer cells are very sensitive for deuterium depletion. Recently, we repeated the in vitro study. We used the XCELLigence system to follow the cell growth. In that case, the cells are going in a gold layer, and the only thing we have to do, following how the impedance of the gold layer is changing. So this is the results we have. So we prepared media with a lowering deconcentration. It was clear that the fastest growth rate was in 150 ppm, and as the deconcentration was reducing, the growth rate also was reducing. And we also could prove that slightly increasing the deconcentration over 150 ppm, 200, 300, that way we also could stimulate the cell growth. And in the next study, we asked how sensitive the cells are for deuterium depletion. So in that in vitro study, we changed the deconcentration with 0 ppm, 1 ppm, 2.5, and 5 ppm every 8 hours. And as you see that the green light is different from the red light, which means that the cells recognize 1 ppm decrease of the deuterium in every 8 hours. If we talk about the cell growth regulation, we know we need a green light and we need a red light. This is how the cells can regulate the cell growth. So how did we explain our results? It is well accepted that before the cells start to multiply, a growth hormone binds to the membrane that stimulates the sodium hydrogen antiport. That cause a slight increase in the pH because the cells are excluded the hydrogen. A paper was published in the Nature. In that paper, they published that they isolated the ATPase gene from the yeast. They used that gene to transform this animal cell line, and they could prove that the ATP is working properly in the membrane, secreting the hydrogen ion, and they also could prove the change in the pH. But the surprising result was that that cell line caused tumor in nude mice and caused tumorous phenotype in vitro. So the question was how possible that a single gene, the APTase, became tumorous a cancer cell, a cell line. Our explanation is that the point is when a growth hormone binds to the membrane and activates the hydrogen exclusion system, that will prefer to eliminate the hydrogen. It means that the deuterium hydrogen ratio will increase. And the signal for the cell is not the changing the pH. The signal is the increasing DH ratio. And this is which can turn the cells and start the multiplication. So when the cells were growing in a deuterium depleted media, they were not able to lift up the DH ratio to the threshold, or it took longer time. And this is the reason that we could reduce the growth rate of the cells in deuterium depleted media. The one question whether it can occur. There is a paper which says that the deuterium is not a substrate of the ATPase, which means that the enzyme is so sophisticated that discriminate between the hydrogen deuterium. So in a membrane transport system, they will prefer to eliminate the hydrogen, and that can cause an increase in the DH ratio. But where is the brake system? The brake system is in the mitochondria. When we are eating the food, eat the food, and burn the food to carbon dioxide and metabolic water, that water is deuterium depleted. So everybody, every day, synthesize about 0.3, 0.4 liter deuterium depleted metabolic water. The question how depleted this metabolic water depends on what we eat. Because when we are eating carbohydrate, the carbohydrate will produce metabolic water with 150 ppm, which is close to the natural water. But the more fat we eat, the lower will be the deconcentration of the metabolic water. And the final deconcentration of the metabolic water depends on the calorie intake, ratio coming from the carbohydrate and the metabolic and the lipids. So one conclusion that in a healthy cells, the membrane transport can increase the DH ratio, but because the healthy cells are healthy cells, it means the mitochondria can work properly. That mitochondria is synthesizing, producing deuterium depleted metabolic water. So these two processes, the green light, which belongs to the membrane, and the red light, which belongs to the mitochondria, they are balanced. But we know that there is one common feature of cancer cells, that the mitochondria doesn't work properly, which means that in the cancer cells, the mitochondria is not able to produce deuterium depleted metabolic water. And this is the key issue, that the cancer cells can grow very fast because no breaking the system, which will be able to produce and keeping the deconcentration at a low level. What happened when we intervened and we modified the deuterium hydrogen ratio? So we made one experiment that the mice was treated with the carcinogen agents, and within 24 hours, we could see that the expression of the c-MYC HRAS P63 increased. That happened in the mice when the mice were consuming normal water. When the mice, even they were treated with the carcinogen agents, but consuming deuterium depleted water, that way we could keep on a low level the gene expression. We tested on a couple of genes, but let's test a couple of hundred genes which can be involving in cancer development. So we used a nanostring technology, which allow us to count one by one the copy number of the different genes. In that experiment, the cells, lung cancer cell lines, were kept on a deuterium depleted media, 40 ppm or 80 ppm, normal 150 ppm or 300 ppm. And we tested two sets of genes, 236 genes were cancer-related, and over 500 genes are kinase genes. We investigated only those genes where the changes of the expression exceeded the 30% and the copy number were over 30. So what we found that in over 200 cancer-related genes, 77.5% changed over 30% when we modified the deuterium concentration. And over 65%, the copy number was higher. So finally, we investigated 125 genes where the expression changed over 30% and the copy number was higher than 30. And the number with the kinase genes was 133. This is what we have. There were a couple of combinations, but the point is we see only 3 combinations. There was only one gene which was upregulated in deuterium-depleted media and upregulated in deuterium-enriched media. And we had 5 genes. This was downregulated in deuterium-depleted media, but upregulated in deuterium-enriched media. But almost 90%, 95% of genes were upregulated at 300 ppm. And we had almost extremely the same situation. So what we found that 99% of the genes respond to the higher deconcentration. What is the conclusion? It means when there is a changes in the DH ratio, that will stimulate most of the genes which are responsible for the higher DH ratio. And if we consider our conclusion 1, that all the signal we have known that changing the pH and the expression of given genes is modified and regulated with the higher DH ratio. So that would be the key mechanism how the cell can harmonize the expression of hundreds of and thousands of genes, and that way can be harmonized the metabolism. Can we prevent the cancer keeping the concentration at a low level? So we repeated the mice study with 48 mice, 24 consumed normal water, 24 deuterium depleted water. And we found that after 150 days, the tumor appeared in the control group, but we lost only 2 mice within over 300 days in the treated group. We are very proud that we have a very first anti-cancer drug registered for veterinary use. That was registered over 20 years ago. It means when the vets are using our drug and the dogs and pets and cats consuming deuterium-depleted water, at 50% of them we can cure them. Right now, we are going to start a new study injecting the deuterium-depleted water into the tumor and the surrounding area. And we have tested in a pilot study, and this is what we can see. Here is a hemangiosarcoma on the neck on the animals after the conventional therapy. And when we started the therapy 4 weeks later, 10 weeks later, 14 weeks later, and the dog became completely tumor-free. One reason that the deuterium depletion caused necrosis, that was published from the Karolinska Institute, that the lowering the concentration will cause an oxidative stress, radicals will appear, and this is the reason that the deuterium depletion triggered apoptosis. But we also tested the deuterium depletion in a phase 2 double-blind clinical trial. We randomized prostate cancer patients. We evaluated 44 patients. The study lasted for 4 months. The treated arm patients consumed 85 ppm, the placebo 150 ppm, and everybody received the conventional therapy. Within 4 months, we could see that there was 80% decrease in the tumor marker value, which was only 47% in the treated arm, placebo arm. We could find 160 cube centimeter decrease in the prostate volume, which was only 54 in the placebo arm. But more importantly, we checked the survival after a year. The study lasted only 4 months. We lost 2 people in the treated arm and 9 in the placebo arm, which was quite significant. We have a product on the market, which is called Preventa. This is a deuterium-depleted drinking water, and we have 125, 85, 105, 85, and go down to 25. We have sold about 5 milliliter deuterium-depleted water, and we had a chance to follow thousands of cancer patients who were consuming deuterium-depleted water. I will show you on a couple of slides. So making a homogeneous population from a lung cancer population, and we checked the median survival time. Evaluating 304 lung cancer patients' median survival time, it was 48 months, which means integrating deuterium depletion to conventional therapy, we can get a 6-fold increase in median survival time. And why? Here is just one example with a small cell lung cancer patients with a brain metastasis in 2001. Her life expectancy was about 2-3 months. That was 3 months, 23, 43 months later, and finally she died 12 years later. We also had a chance to follow 204 cancer patients who started to consume deuterium-depleted water in remission. In our database, we had 2,222 cancer patients, and 204 of them started to consume without tumor. They were tumor-free after the therapy. The cumulative follow-up time of these 200 cancer patients was over 1,000 years. The average time to start DDW consumption was 325 days. It means within 1 year after the diagnosis and the therapy and became tumor-free, they started to consume. The average length was over 500 days, and we lost 13 patients within over 1,000 years. 2 patients' cancer death wasn't related to cancer, and 8 patients were lost several years after they stopped consuming deuterium-depleted water. So we concluded that consuming deuterium-depleted water after the therapy and being tumor-free, that way we can prevent the relapse of cancer. So our conclusion, that deuterium-depleted water consumption can prevent the relapse of cancer patients. Integration of deuterium-depleted water into commercial therapy can drastically reduce the cancer-related deaths. The proper dosage and proper protocol for the supportive therapy application of DDD will increase the efficacy of the commercial therapy and can reduce the relapse. And we have numbers for Hungary. We estimate that we can reduce the cancer deaths approximately 60–70%. In Hungary and in worldwide, I can say hundreds of people took part in that type of research. I would say thank you, everybody, and thank you for your attention. You can find my email address and my telephone number. Thank you for your attention.
Thank you, Dr. Gábor, very much. I am aware. My colleague is Dr. Petra Dorfman, and I know Dr. Laszlo very well. So they have introduced. I've just known about deuterium depleted therapy like last year, and of course, for me as an oncologist, in the integrative oncology realm, it has been like an amazing intervention. And can I ask you, like, with the Preventa water, etc., because I am trying to have my patients start their DD water in addition, of course, to the metabolic therapies, or if they're having like low-dose chemotherapy, etc. So can it be like, is it affected by electromagnetic waves, like being transported at airport, etc.? Would it still be like effective?
No, I guess so. The water what we are producing, you are using a fractional distillation, and we following the deconcentration with the laser equipment. And if the water is in a closed bottle, the deuterium level will not change. And anybody who consume it, immediately the deconcentration will reduce in the body, and that will trigger the different effect, and then hopefully triggering the necrosis and the apoptosis of the cancer cells. So I don't believe that there is any effect or need any effect to enhance the efficacy of deuterium depletion. The lowering deconcentration is a big challenge of all cells, for the healthy cells also. But because the metabolism is normal in the healthy cells, they can manage the changing deconcentration. But the cancer cells cannot do it, or it takes much longer time that the cancer cells can adapt to the lowering deconcentration.
Yes, it was very shocking for me to know that healthy mitochondria actually produce deuterium-depleted metabolic water. So this is just amazing. Another thing to add to the miracle of the human body. So I was wondering if we, like, there are certain foods that actually can enhance the metabolic, lowering the deuterium in the body. And what is, if someone has cancer or an autoimmune disease, what is the procedure of applying the deuterium depleted water to them? Because we won't be using the lower one at once, right? So we just gradually decrease the, you know what I mean? Like using it in a gradual manner.
So the Prevent and the product is available. People can buy it, and then we sold it 50 different countries worldwide, and we have distributors in different countries. And one of our last paper was published in the Journal of Cancer Therapy, and we published the data about the lung cancer patients. And in that paper, we wrote a very accurate protocol, which deconcentration, how many months you should consume for one type of water, and when you change for a lower one, just to prevent the adaptation of the cancer cells. And of course, we need lots of work to figure out all the details, which would be the best combination and the best dosage for deuterium depletion, how to combine it with the commercial therapy. But I guess the basic information is available. And the other issue what you asked, the food. So we know that the ketogenic diet was tested and it was very effective to treat different type of cancer. But when they started, they didn't know why it can be good, the ketogenic diet, to cure cancer. And the reason is that the ketogenic diet means deuterium depletion, just because when the water is synthesized 90% from the lipid, that will produce very, very depleted metabolic water. And that means that the deconcentration of the body will be on a very low level. And that makes more difficult for the cells to trigger and to reach the DH ratio, which is necessary to start the cell multiplication.
Amazing, amazing work. I'm a big fan.
Thank you.
I have a question too. So I'm the next presenter, and I have a question. I'm a neuropathologist, and I saw your picture of that lung cancer met, and my question is, did that patient receive in the same time radiotherapy to the met?
Yeah, sure. Everybody.
Okay,
yeah.
So
yeah.
And do you have a control study with lung mets and brain mets? I'm a brain person. Do you have anything that's controlled, showing in a control arm and in a treated arm with lung mets?
So the only prospective study we could complete, that was a prostate study. That was randomized, double-blind clinical study. All the data, or most of the data what we were able to publish is based upon the retrospective study, and we are comparing to the historical control. And just because when we're comparing the median survival time, we can get a 3 to 10-fold increase. So it's not only 10% or 20% increase. And it is so big differences that I could say that this is due to the differences that these people receive the conventional therapy plus consuming deuterium depleted water.
Why I'm asking is that brain is a little particular because it has the brain-blood barrier, the BBB, and many drugs do not penetrate it. So it would be important to see if these meds really regress because of also, it would be an addition to the conventional therapy. It would be interesting because it's not only meds, but I'm going to present next, it's also glioblastoma. And a prospective study can be done pretty easy because the survival is very dim, like it's 1 year. So you could see immediately what if there's changes with that.
So first of all, based upon the basic rules of the nature, deuterium depleted water appear in the brain. It cannot be any other way because the water is moving. And there are studies when they used heavy water and they could detect the increase of deuterium level in the brain applying deuterium, heavy water. This is one thing. The other, we also have a couple of data with glioblastoma, and we also could see how we can increase the median survival time or we can prevent the relapse. And the only question is, how can we get $5 million to run a phase 2 clinical trial? So it is not the lack of our intention to do more clinical study. This is a lack of the funding. And
yeah, sure. Like
the
always.
only reason that we could proceed that we are selling the water, that way we could finance our drug development. And this is where we are now, and we are waiting for the investors. And I do hope that investors will come, and then we will complete lots of several clinical study proving that and register it as a drug.
So I have a second question. This comes from my previous work in poliovirus. So it's a paper, long time when I was in Pasteur Institute, showing that the deuterium water can stabilize, thermostabilize the poliovirus live vaccine. And do you have any idea? Looks like it would have also an additional effect on the protein itself, something like I don't know, kind of coating or something like this. Like, is that something that you're also studying, some protective?
Yeah, so I think and this is the amazing, when the deuterium hydrogen ratio is changing, it means that each molecule which has hydrogen in the exchangeable position will be a new equilibrium. And when we modify that, the protein structure can be more flexible or maybe more rigid. And the DNA, RNA stability can be different. And so that way, all the molecules will receive a message when the DH ratio is changing, and that will reprogramize the whole process in the cell. And that happened with all the molecules. So not just targeting one gene and one tyrosine.
Right. It's not only, it looks like, I mean, everything, like also proteins, they have, I'm more of a, I mean, I'm everything, but I worked with proteins a lot, and they do have hydrophobic and, you know, and hydrophilic regions. And it looks like this water, heavy water, somehow modifying the, you know, the hydrophobicity, so it has a more, you know, larger impact than on you know, redox and all that. It looks, I'm not sure, but that's what we showed back then, that it thermostabilizes the poliovirus, which is a particle in deuterium water, in heavy water. So it's very interesting.
Thank you.
One last question, please. Is there some sort of, I've heard about, like, for instance, I cannot get Preventa to Egypt, but there are certain nutraceuticals, or is there a way to create like a DDW in, like, at home, or certain nutraceuticals like curcumin? I think it actually improves the DH ratio.
So no, our facility is about 12 meter high. The other facility is 30 meter high and needs your tons of steam to produce a couple of liters deuterium depleted water. It's not so easy. But I don't believe that you cannot buy. So it's a question of, so someone order, we can deliver it.
No, it's something about the customs to get it out from the airports. Yes,
it's okay. It could be difficult. Yeah.
Thank you very much.
Thank you.